Now I'd like to introduce Lorenzo Leggio, who was kind enough to provide an introduction for Dr. Hanlon.
Okay. Thank you, Tom. So it's really my pleasure to introduce our first speaker today, who is Dr. Colleen Hanlon from Charleston. Dr. Hanlon is an associate professor at the Department of Psychiatry, jointly with the Department of Neuroscience at the Medical University of South Carolina. And she's also the scientific co-director of the Brain Stimulation Division at the Medical University of South Carolina. Colleen received her Ph.D. from Duke University. Then she moved to Wake Forest as a postdoc fellow. And after a brief junior faculty appointment as an instructor, she moved to NUSC as an assistant professor, where she was on faculty as assistant professor for 5 years, funded by NIDA through a K01. And then when the K01 was over, she was promoted to associate professor, which is her current faculty position, together to co-direct the Brain Stimulation Division. And she received several grants, primarily from NIDA and NIAAA. Recently, actively, she is a PI in a NIDA grant R01. She's also PI in another NIDA R21. She's also a PI of a component of the NUSC Charleston Alcohol Research Center, which is funded by NIAAA, and she's also a project leader in a P20, which is investigating the neurobiological basis for loss of cortical laterality in chronic stroke patients. And Colleen has many, many achievements, including she has already published 44 papers, 50% of them as either first or last author. She has received many awards. You will forgive me to not mention all the 2-page awards from your CV. I will just mention 2 from ASLBP and ACMP. She also serves in the editorial board of the Experimental Clinical Psychopharmacology Journal. She's an associate member of ACMP. And in addition to her research, she does a lot of teaching at MUSC, where she has already established herself nationally and internationally for her expertise on neuroimaging and brain stimulation. She has given 60 talks, both nationally and internationally. And so to summarize, basically, Dr. Hanlon's overall theme and scientific research has been to map neural circuitry irregularities in alcohol and substance use disorder patients, and then modulate these circuitries using brain stimulation techniques or neurofeedback. So with that said, join me in welcome Dr. Colleen Hanlon. The title today is going to be From Mapping to Modulation, Using Intrinsic Neural Architecture to Develop TMS as a New Treatment for Craving and Control. Colleen, thanks so much for being here, and we look forward to your talk.
Thanks so much. Well, thank you all very much, Glenn. Thank you for such a nice introduction. That was great. I'm extremely happy to be here. Let me see if I can get my talk started. And today, first, actually, I want to thank of NIDA and NIMH and NIAAA and whomever else, NINDS, sort of helped organize this. I know the neuromodulation unit here at NIMH has organized this great symposium, and I hear that hundreds of people are potentially watching online, which I'm sure doesn't make me the slightest bit nervous, but I'll do my best. And anyway, I really want to say that it's great to see all this enthusiasm from everyone about really trying to develop TMS as a rigorous, evidence-based treatment for various psychiatric and neurologic disorders. What I'm going to talk to you about most today is about addiction. So addiction can be seen as a disease of both craving and loss of control. And that will be a conceptual model and theme that kind of falls through the talk. So the sort of obligate conflict of interest slide, all of the work that I've done has fortunately been funded through the NIH, and I'm very thankful for that. And there's a ton of people that have contributed to this work. I just happen to be their ambassador. So to start off, what do we know about the brain? Well, there's many models of neural circuitry. Certainly in the last decade or two, we have had significant advances in our understanding of fronto-striatal circuitry in the brain. We know that there are many dopaminergic circuits in the brain, one of which is the mesolimbic dopamine circuit. This is supposed to be involved in motivation, vigilance, sometimes this is called hot cognition. Additionally, we have the mesocortical dopamine system. This is more involved in executive processing, response inhibition. Sometimes this is referred to as cold cognition. And then we have the nigrostriatal dopamine system. And although I spend a lot of my time thinking about the mesocortical and mesolimbic tone, we do in the Department of Psychiatry, if I were to walk across the street to the Department of Neurology or to NINDS, they in fact spend a lot of time thinking about the nigrostriatal dopamine system. And of course we know that these dopaminergic circuits kind of go through preclinical and clinical models. Suzanne Haber and Brian Knutson have done a really elegant job in a review article in Neuropsychopharmacology about non-human primate track tracing work of these circuits. Another famous paper by Alexander DeLong and Strick is sort of told us that these are parallel and integrated circuits. This set up a lot of work in the field. And then a more recent paper in Nature Reviews Neuroscience was also very well done. And again, it mimics this same idea of these parallel and integrated frontal striatal circuits and their relative control over behavior, one of which is more associated with limbic tone and drive, another is executive control, response inhibition, and then another one is, in fact, you could argue behavior. So all of behavior requires action. And so we probably should not neglect within psychiatry the role of the nigrostriatal dopamine system. What do we know about cocaine? Well, we know that cocaine directly affects dopamine in the striatum. Of all of the substances of abuse, cocaine is one of the few that directly affects dopamine and causes a large change in dopamine binding. Of course, methamphetamine is potentially even more efficient in this way. Well, my postdoc advisor, Linda Perino, demonstrated in a non-human primate model that over time, when you use cocaine, the effects of cocaine initially begin in the ventral aspects of the striatum, which are the more limbic domains, and then over time, the effects grow out to more sort of dorsal, lateral aspects of the striatum. This is true not only in changes in dopamine, but it's also changes in glucose consumption, and other measures of excitation. We then showed that in humans, individuals that are chronically using cocaine also have these changes in the dorsal striatum. And in fact, in individuals that have been using cocaine for 15 or more years, you lose the typical functional segregation between the ventral and the dorsal striatum. Again, as sort of neuroimaging techniques have grown through a lot of the work that's been done here at NIH intramural, we now know that there's all of these sort of functional connectivity networks that exist in the brain. And through the addiction process, this is a figure that I pulled from a recent very good review by Powers et al. That shows that the engagement of various baseline functional connectivity circuitry in the brain changes over the course of the addiction process. So to those of you that are not addiction researchers in the audience, addiction is seen as a progressive disease, one that behaviorally starts with a vulnerable individual who's given sort of opportunities to enjoy the reinforcing qualities of the substance like cocaine. And initially, drug-taking behavior is mediated by reward-based mechanisms. But then over time, there's an evolution, and drug-taking behavior becomes much more habitual, much more behaviorally and ritualistically driven. And what this figure shows here is this cascade from incentive craving and goal formation, which typically occurs in the early stages of the addiction process, eventually to action and habitual control. It involves different stages of neural architecture. So as you can see, there's now a lot of research on functional imaging, functional connectivity, and the role of these different cortical, subcortical networks in different stages of addiction. Well, it's one thing to spend a lot of your time, like I did for most of my career and still do, just mapping the brain, just looking at these individuals and saying what's different with their brain. But it's even more exciting to try to change that behavior. And now, in the last few years, we have a number of studies, which you can see here, that have used functional imaging in individuals that are undergoing rehab for various substances. So this is a summary of all the studies that we could find as of last year that have looked at baseline brain activity to either an executive control task, those are the ones in blue here, or to a limbic arousal task, and looked at the ability of their brain activity at baseline to predict their treatment outcome. And what you can see here in this summary figure is that when engaged in an executive control task, the areas in blue, if they have decreased connectivity in those areas, individuals are more likely to relapse. Individuals that present with relatively high connectivity in the red areas also have an increased ability to relapse. And so that suggests that potentially there's a predictive role of baseline brain activity to eventual treatment outcome. Well, that sets up stuff really nicely. The preclinical groups have made tremendous strides in the last decade or two with really elegant molecular tools where they can causally manipulate these circuits. And what many of them have shown, here's a sample of some of the papers that have been particularly influential, is that through optogenetic stimulation of the prelimbic versus the infralimbic cortex, you can, in a causal manner, increase or decrease cocaine self-administration. So what that means is these same brain regions, the ventral medial prefrontal cortex, orbital frontal cortex, nucleus accumbens, all these limbic, cortical, subcortical connections have a causal role in drug self-administration. And so we, as human researchers who are looking to really translate these data into something we can actually deliver to our patients, would really like to see if we can do the same thing in humans. Can we take TMS and in the same way causally change activity in these networks and then change eventual treatment outcome? So that's pretty much the goal of my lab for the last few years, and hopefully for the next few years to come, is to develop TMS as an evidence-based neural circuit-specific treatment for cocaine addiction. We're getting close. And I'm going to tell you, and then for the rest of the talk, about logical progression. We've done at least 6 studies which logically have followed one another and have led us to a point where we're now running a double-blinded, placebo-controlled clinical trial of TMS as a treatment for cocaine users. Okay, so the strategy that we've chosen is to stimulate the ventral medial prefrontal cortex by quieting down this brain region. So that's kind of the conclusion. I'm just going to whet your appetite, and I'm going to tell you how we got to there. Okay, so the general outline for this presentation is to first present you with a conceptual framework for developing TMS as a treatment tool for addiction. It's already an FDA-approved treatment tool for depression, and there's a lot of people out there that are trying to optimize various parameters, etc. But in addiction, we're way behind the depression crowd. We're probably way behind the pain crowd. But we have a lot of really good preclinical evidence that actually can help lead us in the right direction. So first I'm going to talk about a framework. Then I'm going to talk to you guys a little bit about some patient-inspired methods development that we've done. Because the audience probably has some baseline knowledge of TMS, I'm kind of going to skipping a lot of the basic TMS stuff I would talk about, but if anybody out there in listener land would like to know a little more about basics of TMS, we have a class, and actually there's several very good classes across the country where we as a community are really trying to teach rigorous methods development for acquiring TMS. And I think that we'll start to see that more in like K01 training plans and F32 training plans. So I highly encourage you to contact me or, frankly, anybody else on the list of speakers coming to this seminar series. And then finally, I'll tell you about what exactly we're doing for our patients. Okay, so again, dopaminergic disorders, typically we think about the mesolimbic system, which is your drive for cues, your drive for drugs or rewarding stimuli, and then your executive control system, which are kind of the brakes to that process. Cocaine and alcohol affect the striatum and therefore change drug self-administration. And so the working hypothesis that we are using in our lab is to say that in general, potentially what causes some people to sort of be vulnerable to becoming substance dependent is that they have a disrupted ratio between the executive control circuit and the limbic circuit. So the way that I really like to sort of talk about this and explain this to people is through an analogy of alcohol, because as a very societally endorsed drug, many of us have had experience self-administering alcohol, potentially more than we should on some occasions, and hopefully we've also had occasions where we have said no in the face of an alternative reinforcer. And so if you think about a time in which you were sort of, let's say, out at a bar with friends and colleagues, and the time was getting late, and then somebody in the crowd says, "One more round," you might think to yourself, "Ooh, oh, that would be so good. It would taste great. I would get socially reinforced for it. It'd be free." That's your reward system, right? That's the red system there. Thinking about all of the positive hedonic aspects of it. But then, hopefully, you have another part of you that says, "Oh, but I shouldn't. I have to drive home. I have to give a talk tomorrow. My significant other will be mad at me." And so you weigh the relative value, the incentive salience of these two circuits. And so it's not bad if sometimes you stay and you have another one, generally, but it is bad if you tend to always stay and have another one. And that's what leads you to sort of DSM criteria for substance dependence. And so what we think is that there's at least two fundamental ways we could think about developing a treatment for cocaine and potentially for alcohol as well. One option, of course, is to just decrease the limbic arousal. So in the presentation of a line of cocaine or a crack pipe or just another drink, the relative value of that reinforcer is lower, and so you don't seek it. The other option is just to increase your ability to resist. So in some ways, the incentive value or the salience of the reinforcer may remain the same, but your ability to walk away from the system is much stronger. And if we think about those two general ways to conceptualize it, we could use TMS, which has frequency-dependent effects on cortical excitability, as a tool to either increase activity in this executive control circuit through something like 10 hertz or 20 hertz stimulation or intermittent theta burst stimulation, or we could try to decrease activity in this limbic arousal craving circuitry with LTD-like stimulation, something like 1 hertz, 5 hertz, or maybe continuous theta burst. Well, if you look at the number of studies that have been done for RTMS and addiction, and I apologize in advance, it actually used to be really easy to keep this table up to date. This is a review article we published in 2015, and now it's actually exponentially increasing. And so thank you all out there for your interest. It's awesome, and I apologize that your name doesn't appear on the slide if it's not here. But in fact, that's a really good sign. But if we just kind of take a snapshot at about 2015, if you just sort of look down this column, most places say DLPFC, 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 DLPFC. Well, that's great. So the left dorsolateral prefrontal cortex, some people are doing their right dorsolateral prefrontal cortex, seems to be overwhelmingly the site that people are trying to treat. It also happens to be the site that we treat for depression. Right? So the standard clinical treatment for depression is approximately 10 hertz given over the left dorsolateral prefrontal cortex, maybe sometimes the right, for several weeks at a time. Well, that's great. We have a big brain out there, and part of me just wants to explore other places. So there's been a lot of great work coming out of Toronto for the last few years. Stanford groups are doing a lot of good work now where they're exploring other aspects of the cortex. Most of these studies are still being done in depression because depression is really leading the field. But in fact, there's a lot of arguments that for addiction, we should maybe stimulate the limbic circuit directly. So we are interested in trying some more of the medial wall studies with keeping our eyes open to the fact that a lot of people are already getting pretty good data with dorsolateral prefrontal cortex. Pejoratively, sometimes I like to describe the dorsolateral prefrontal cortex as the magic brain button because it seems to sort of cure everything with RTMS. And you know, I say that sort of tongue-in-cheek, but it may actually be true. But anyway, we'll see as the field emerges. And so my favorite scientist, perhaps, Sherlock Holmes, said, it is a capital mistake to theorize before one has data. Insensibly, one begins to twist fact to suit theories, instead of theories to suit the facts. And so with this, I think we really, especially now, we need to be evidence-based, and we need to explore the neural circuits. We have these great tools, functional connectivity, to do mapping. We have task-based sort of neuronavigation, and all of these tools will point us to sort of a smarter answer than just doing what everybody else does because it seems to work. Okay, so that has been our goal, and we have had 6 sort of logical steps to sort of get there. When thinking about the possibility of stimulating that red circuit, the craving circuit, the first resistance that I had was from people that said, well, there's no way that you're going to be able to hit the ventromedial prefrontal cortex with TMS. Your frontal sinuses are in the way, and the magnetic field decays very, very fast. So we thought about that, and we addressed it. And then the next idea was to say, well, is it even possible to try to distinguish this red circuit from this blue circuit? Can we actually even parse them apart and be smart about stimulating them? And once we did that, we wanted to say, okay, now that we have these 2 circuits, are they different in cocaine users? Because we needed to know where to treat. Should we go to the dorsolateral prefrontal cortex, like people have been doing, or maybe should we go to the ventromedial prefrontal cortex? Once we found an answer to that, we wanted to evaluate if it was possible to move the circuit. So in just a single day, could we induce transient LTP or LTD-like effects in the circuit that we chose? This is connectivity between the frontal cortex node that we're stimulating and the subcortical node. And then finally, once we figure out that we can move the circuit around in a single day, we can evaluate whether or not it's feasible, safe, and if we have efficacy and durability of long-term TMS treatment. Because again, for the depression treatment protocols, you stimulate for at least 6 weeks, typically, and that's when you start to see behavioral change. There's a lot of reason to believe that in addiction, particularly stimulants, it might not take that long. The preponderance of evidence in pain research suggests that you probably need about 10 days or 15 days. So shorter is probably possible, but we just wanted to see. And then finally, if I have time, I'll talk a little bit about the area that I think a lot of work is going on now, and a lot of the other seminar speakers are actually have more expertise in this domain, is the role of individual variability. So in this sort of spirit of precision medicine and being able to choose individuals or states or sort of baseline cortical excitability states through EEG rhythms that might optimize treatment efficacy for our patients, what should we consider? Okay, so basic TMS 101, stimulation depth. So with a standard figure of 8 coil, the general dogma is that it takes about, standard figure of 8 will penetrate about 2 centimeters, or 20 millimeters deep at motor threshold. But generally the excitation is limited to the cortical mantle. However, if you can get to the cortex is only about 1.5 to 3 millimeters thick. Cortex is very thin. If you just take a moment to think about that, everything that makes us human is only about 1.5 millimeters thick. Okay, so you think that's actually phenomenal, and it's worth thinking about. And so our idea, knowing these facts, was to say, okay, well, we just need to get to the cortex with TMS. And then once we get there, we think we can probably penetrate all 5 layers and do what we need to do on a cellular level. I just kind of to sort of add to the group, recently I was speaking to a reporter, and this person said, well, when you put a TMS coil on the brain, about how much of the brain are you stimulating? And so I said, oh, 2 centimeters. And she asks, well, if that was a map of the United States, about how much would you be stimulating? Like a region or a city or a county or a state? So I thought, oh, well, that is a testable question. And so I ran back to my office and I ran the numbers, and sure enough, TMS, approximately 2 centimeters of stimulation, the surface area of a coil, is approximately the great state of South Carolina, if you were to put it on a map of the United States. So now maybe you'll never forget that because I said it. And so that's a really good visual, right? So that's about how much, if we just remain very America-centric, then we can stick to about the state of South Carolina. I just published this as like a little letter to the editor in Brain Stimulation, and I included some other like countries in the little diagram if you want to see that. Anyway, so that's kind of fun. Okay, but back to science. So this was actually inspired by reviewers, because in fact reviewers actually tend to always know what they're talking about. And these reviewers were very concerned about the depth, the distance from the cortex to the scalp, particularly in individuals with cocaine and alcohol use disorders who had, a lot of, they're using alcohol a lot. There's a lot of evidence to suggest that alcohol changes gray and white matter density associated with atrophy. And so they were rightly concerned that maybe we wouldn't be hitting the cortex. And so we developed a tool to sort of automatically unbiased calculation of the distance from the scalp to the skull. And as you can see here, if you look at F3, this is the DLPFC position, and FP1, this is the frontal pole position. Actually, the distance is pretty close. So the strength of the magnetic field falls off very quickly with figure of 8 coils. It's maybe 1 over a cube root. And so it's very fast, and every little bit of distance matters. But thankfully, the distance isn't too different. And this is just an image, again, of a representative control and a representative cocaine user and how we measure the distance. This is a cocaine user here with obvious atrophy and sulcal enlargement. So if you're not used to looking at heavy drug users, this actually looks fairly similar to the kind of brain you would see in an alcohol user. You see these very large sulci, right? And so this atrophy, though, in this particular individual still was kind of preserved. There wasn't very much atrophy at the level of the frontal pole. And so we were fairly convinced that in this guy, we still sort of hit the cortex with the TMS coil. But these are really important parameters to consider because the strength of the field falls off so quickly. And so we now, as a laboratory, have begun to include scalp to cortex distance as a regressor in all of our analyses. And I could definitely see that in general, not only would we use them in the future as regressors post hoc, but we could use them as dosing metrics pre-hoc, right? So you could figure out the dose of TMS that you need to give based upon their scalp to cortex distance in a various spot. Of course, this isn't an issue if you're studying motor control because your dosing metric is your MEP. But again, just because the motor threshold is at a certain value, that doesn't tell you how much the frontal cortex the distance is going to play a factor. And so an outstanding engineer that used to do preclinical work with me is Philip Summers. He did preclinical work with my colleague, Andy Sheet, and he was looking at vessel dilation using 2-photon spectroscopy. And he developed a tool for us to calculate a surface fit and the volume of CSF that are present in various locations. So this tool is called Brain Ruler, and it's free, open source, and available on MathWorks. So you can download it. It runs in MATLAB. It's fairly easy to use, and it has a GUI interface, which is nice. Okay, so then another thing we wanted to figure out, specific for our patients in developing an evidence-based treatment for these cocaine users, was can we actually get to the subcortical brain regions if we choose to stimulate the DLPFC or if we choose to stimulate the ventromedial prefrontal cortex? So we know from a lot of great work that's come before us that TMS induces activity at the site of the coil as well as monosynaptic striatal afferents. And there's been a number of very elegant work studies that have been done. Some of my favorites are by Straffella's group, and they've shown that with different frequencies of stimulation, you can change dopamine binding in the striatum with cortical TMS. So this is great. When we moved again to the prefrontal cortex, these things become a little bit harder. So the dorsolateral prefrontal cortex is, again, the FDA-approved treatment for depression. And Strifello's group also did some dopamine binding studies in looking at the effects of DLPFC stimulation on striatal dopamine, and he saw a frequency-dependent change. But we weren't so sure about the medial prefrontal cortex. Now, again, theoretically, it should follow, but we have all of these other issues and constraints that might happen. In fact, we weren't even necessarily sure that if we put a coil on the frontal pole right here, that we would actually be able to activate the striatum. It just hadn't been done. Additionally, we were wondering, a lot of studies came before us that used TMS in the MRI scanner to sort of apply a pulse at the level of the cortex and watch what happens at the level of the striatum. And they would see changes in bold signal in the striatum, but they would also see changes in bold signal in the cingulate, in the motor cortex, in the temporal lobes, all over the place. And of course, as good scientists, we want to make sure that that's a causal change and not just related to the other aspects of receiving TMS in the scanner. So if you've ever had TMS yourself, just single pulses, you know that it's a little weird, especially if it comes at an unexpected time. It can be associated with movement, even if you put it over the DLPFC or the medial prefrontal cortex, because you kind of want to move away from the coil. So there's all of these sort of human subject factors. And the field is generally very good and very rigorous about insisting that everyone in the field use good active sham controls. And yet, really, we don't insist that people that do interleaved TMS fMRI have kind of a sham control condition, myself included. So an outstanding graduate student in my lab, Logan Dowdle, sort of set out to kind of take on this task. The coil, just to tell you a little, so MUSC was the first place in the world to take a TMS coil and put it into the MRI environment and apply pulses. This is done by Mark George. He's the director of the Brain Stimulation Division at MUSC and my boss. And he did a lot of this pioneering work. He was actually in this building where he did a lot of his training before. And so he, at that time, developed this coil holder here. So this is an early TMS coil. This is Dantec, who was later bought out, I believe, by MagVenture. And this Dantec coil is actually really nice and light and small. And this is the coil holder. So you can take this TMS coil and you can position it wherever you want over the person's head. It actually will go all the way back to the parietal cortex. And the secret to this is our coil holder here. So this is our TMS targeting system. I know that some of you are thinking about developing TMS, interleaved TMS systems, and we're happy. You can just call us or email us, and we're happy to tell you what we know. Okay, and so of course, you know, it's always nice to sort of see the data yourself, right? And so what Logan and I did is we took the TMS coil and we put it over the dorsolateral prefrontal cortex. We wanted to see if, in fact, we could get a bold signal. And I, at the time, being relatively new to TMS, thought, well, clearly, we will get a bold signal underneath the coil, because that's where we're activating, and we will get a bold signal in the striatum, because that's the monosynaptic afferent. Well, turns out that doesn't actually turn out to be true. So what Logan did is he placed the TMS coil here on the dorsolateral prefrontal cortex, and he acquired data very fast, so at a TR of 1 second. Because we had the full story is we had actually tried this already, and we weren't seeing bold signal underneath the coil. So we had a hunch that maybe it was because the hemodynamics underneath the coil were different than the hemodynamics at the monostriatal afferents. The way that TMS works, the mechanism of action, is really at the level of the axon rather than the initial cell-cell body. And therefore, the bold signal at the area that's directly stimulated is likely not the same as the shape of the bold signal in the striatal regions. So when you just naively do this and you run your data through FSL or SPM or AFNI, which assumes a canonical hemodynamic response function, you're not going to see anything convolved with that. Okay, so we did high resolution. So we used a 1 second TR, and we fired the pulse within a gap between each measurement or each volume that's collected. We had 20 TMS pulses per session, and the inter-TMS pulse interval was 10 to 15 seconds. And this was because we wanted enough time to see the bold signal rise and fall nearly completely. So for those of you out there that are neuroimagers, this is like the world's slowest event-related design. And we modeled it that way. And what Logan found, and hopefully this will be submitted soon, is that when you apply TMS to the dorsolateral prefrontal cortex and you have sham stimulation, so in this case, sorry, I forgot to add this, that his sham condition or his control condition, in this case we exploited the idea of distance from the scalp to the cortex as our way to create a control. And so we took open cell reticulated foam and we pressed it down such that it would convey the mechanical force associated with the TMS coil, but it would be far enough that the magnetic field wouldn't penetrate the foam and get into the cortex to cause excitability. So the sort of sensory aspects of TMS in the scanner would be consistent, but the sort of direct TMS effects would not be, would be able to be isolated. And so what Logan showed here in this percent signal change graph is this is the percent bold signal change on the y-axis, and this is time after the bold signal on the x-axis. And so as you can see here, if you're familiar, the dotted line here is sort of a typical hemodynamic response function. The dotted line is the hemodynamic response function that was associated with the sham. So in fact, sham itself caused a bold signal in the striatum. But the bold signal in the striatum associated with real stimulation was greater. And it was greater, the difference was enough that we could actually detect it. And so if you look here, he also found that there is a dose-dependent relationship between the amplifier output and the evoked bold signal in the striatum, suggesting that there's a causal link between the amount of energy that's going in and the amount of bold signal that's happening in the striatum. So that got us very excited. And here, I won't spend too much time on this, but hopefully this paper will come out and you can read it. He did a region of interest analysis, and what he found was that of all of these regions of interest, the only regions there was increase in bold signal associated with the TMS pulse in many regions, but the regions in which were differentially activated by single pulses of TMS to the dorsolateral prefrontal cortex were the anterior cingulate cortex and the caudate. So to many of us in the field, that's a sigh of relief, because that's great. Those are the brain regions that we depend upon, the brain regions that we build our conceptual models upon, and probably very relevant to neuropsychiatric disease. Okay, so the next step was, great, so we think that we can actually stimulate the dorsal striatum and that there may be dose-dependent effects on the bold signal. We wanted to know, okay, can we discriminate this sort of mesolimbic from mesocortical, hot from cold cognition circuit? We started with just healthy controls. This is done with a team of people in the lab. We also developed a tool for rigorous positioning of the coil for this particular experiment, and you can find the tool. It's freely available as well. This is done between Jeff Burkhart and I, and basically it's just a nice handy tool to use to sort of find the EEG 1020 system locations for various places that you want to put the coil. So as we expand out to applying TMS to different brain regions, these kind of tools will be increasingly useful to the field. At least I hope so. Okay, and again, and this is the standard TMS bold experiment. Individuals are laying in the scanner, and essentially it's a slow event-related design where a TMS pulse occurs every, in this case, about 10 seconds. And what we find here is that when we position the coil over the dorsolateral prefrontal cortex, we seem to get evoked bold signal on the dorsal aspects of, or lateral aspects of the prefrontal cortex, as well as in some caudate on the ipsilateral side. And when we place the coil over the medial prefrontal cortex, in this case, the frontal pole, the left frontal pole, we get evoked bold signal in the area directly underneath the coil, or maybe a little bit further back from the coil, as well as in the striatum itself. I will say that we've now upgraded to a Prisma scanner at MUSC, and Logan has been sort of leading this project and playing with multi-echo imaging, so both multi-slice and multi-echo. And the data, I actually would love to redo this experiment with those parameters. So I do think that multi-echo imaging probably is going to really advance our ability to look at the brain through interleaved TMS fMRI. Okay, great. So now, we think that we can separate these circuits. We think that if nothing else, by putting the TMS coil over the DLPFC, we can get more dorsal aspects of the frontal-striatal system. And by putting the coil over the medial prefrontal cortex, we can get more ventral aspects of the limbic system. And this is published as well, so feel free to look it up. Okay, so that's great. So now we are pretty convinced that we can apply TMS to the medial prefrontal cortex, the ventral medial prefrontal cortex, or the frontal pole, and get changes in bulb signal. There's now been a lot of groups that have done this, and so it actually seems like sort of obvious now. But at the time, it wasn't really obvious, so we were happy to sort of see it. Okay, great. So now we've just looked at healthy controls. We wanted to see what these circuits look like in cocaine users. And so in this case, this study was led by Melanie Canterbury, a postdoc of mine at the time. And we had 20 cocaine-dependent individuals, all of whom had had failed quit attempts, which is a very important aspect of cocaine dependence, and 20 age and gender matched controls. And in this case, in the same way, we put the TMS coil over the dorsolateral prefrontal cortex and the medial prefrontal cortex. In the controls, and we learned a little bit from our first study. So now we're doing many more TMS pulses. So it does, in fact, take probably at least 20 to 30 pulses to sort of observe a consistent signal. And so here, we apply the TMS pulse to the dorsolateral prefrontal cortex, and we see evoked bulb signal in the controls in the dorsal striatum, as well as in the cingulate. And in the controls, we also see bulb signal in the orbital frontal cortex or ventral medial aspects of the prefrontal cortex, as well as the ventral striatum. So great. So we kind of replicated the same pattern that we observed in the first study, which made us feel good. In the cocaine users, DLPFC stimulation evokes a fairly similar pattern. So you see here activity in both the dorsal striatum and in the anterior cingulate cortex. So that's pretty interesting. And but in the cocaine users, when you put the coil on the medial prefrontal cortex, so you apply stimulation to this brain region, which is typically mobilized by rewarding stimuli or evocative stimuli, the cocaine users have a much lower response. So in a group comparison, you see there's actually not too much that's different between the users and the controls in their brain reactivity to a single TMS pulse when you stimulate the DLPFC. But when you stimulate the MPFC, there is a big difference. And that is the cocaine users don't seem to mobilize this fronto-striatal limbic circuit in the same way that the cocaine users do, that the controls do, I'm sorry. So that was the area that was the most different. And so based upon some resting state data, which also suggested that this circuit between the medial prefrontal cortex and the striatum was particularly aberrant in cocaine users, and that functional connectivity was a little bit too high than usual, we decided to go with this circuit as our target for TMS treatment development. Next step. Okay, so we have a target. We feel like we've been fairly evidence-based getting there. We have a lot of preclinical data to sort of help grow our hypothesis and refine the ways we're going to do it. So now our idea is, can we move this circuit at all? Can we induce just a transient state of LTP or LTD in this circuit? And so we did a study where we were trying to decrease the brain response to cocaine cues. So craving is one of many factors that leads to relapse, but in fact it's one of the most important factors and consistent factors, not only in cocaine users, but transdiagnostically appropriate. And so we wanted to see if we wanted to test two questions. Can we use TMS, an LTD-like form of TMS, to dampen activity in this fronto-striatal circuit? And can we use TMS to dampen cue reactivity? So the way in which the brain naturally responds to these cocaine cues. And so the protocol that we developed is frontal pole stimulation. So this is theta burst stimulation, an LTD-like form of theta burst stimulation. So those in the audience that spend a lot of time thinking about this, you may criticize me for saying that it's LTD-like because it's all pulse dependent, pulse number dependent. But for now, we'll just go with the facts. And the facts are that we use 3,600 pulses of TMS to the frontal pole. It was delivered in continuous theta burst stimulation fashion. There were 2 trains, 1,800 pulses per train, and there was a 60-second intertrain interval. So essentially, we had 1,800 pulses, and then we paused, and then we did 1,800 pulses. The whole protocol can be conducted within 5 minutes. In these studies, we did ramping of the TMS dose, which I'll tell you about in a second. And we call this kind of a sandwich design, where we put them in the scanner, and we showed them pictures of cocaine cues or neutral cues. And then we also used interleaved TMS fMRI to look at their baseline brain reactivity. We then gave them a dose of real or sham CTBS to the ventromedial prefrontal cortex, and we put them immediately back in the scanner. These studies, there were 49 individuals enrolled in the study, and it was a within subject design. So everyone received both real and sham stimulation. So there's a lot of scanning and a lot of data. This is actually what the protocol looks like, so hopefully the sound will work. These data were recently published. Actually, I think it's available now online. So it was just recently published. And we published in the supplementary material our standard operating procedure for this procedure. So I think that's really important, actually, because as I've had pleasure of talking to you guys and to some people at NIDA yesterday, we're all in this together, and we all learn a lot that we never actually really publish very much, because you write everything up. And so the more that we can start, like in our supplementary material, publishing standard operating procedures, or even kind of creating a forum or a community where we can post tips that we learn, I think the stronger that we'll become. So this is published in the supplementary material of our recent paper, and the video is published as well, so feel free to look it up. Okay, so this is Oliver, my brave research assistant, and this is Scott, my other research assistant, and they will be demonstrating what we do for our patients. What you'll see in this video is this is the TMS coil. This happens to be a MagVenture coil that's sham. It's a sham coil, so it's real on one side and sham on the other side, and it's placed over Oliver's left frontal pole. The cords that you see running underneath it are electrodes, and the electrodes are engaged to kind of help mimic the sham in the case that it's sham. In this case, it's real, and you'll see Scott in the background turning a knob. And what Scott is doing is critically important to delivering TMS to the frontal pole. He'll be ramping the dose. So in our case, this protocol, 1,800 pulses followed by a break, followed by another 1,800 pulses, takes about 5 minutes. So we have some time to ramp up the dose versus the standard 600 pulse protocols are very quick. And so delivering them on the forehead can be very painful because you can't ramp the dose. But if you have the ability to ramp the dose, you can get an individual to tolerate up to sort of 110% or whatever it is that you want to do of motor threshold. So in this case, you'll see Scott ramping up the dose slowly, and you'll observe Oliver receiving the treatment. I will say, just for full disclosure, I've definitely also received this on my head many times, and they volunteered freely and willing to do this. Okay. So you see, Scott is ramping the dose in the background. I think I'll zoom in a second, and you'll be able to see Oliver's forehead moving a little bit. So because of the frontalis muscle on the forehead, you'll start to see a little bit of eyebrow movement. So that's Oliver saying that he can kind of feel the difference as an experienced TMS person of the TMS pulses themselves versus the sensation of the electrodes. And now we've reached the dose, so that was our ramping protocol. And then we'll continue for the full, I guess, about a minute and a half for the 1,800 pulses. So I'll stop it there. So that's what we do. And we've done this now on over 100 substance-dependent individuals. We've had no serious adverse events. We occasionally have people that come in, and they'll come in for a couple visits, and they'll decide that they don't really want to do it anymore. But we certainly haven't had like the typical adverse events that you worry about with our TMS procedures. So we feel good. It's always good to be cautious, and so we're always cautious and watching and monitoring. But patient compliance seems to be pretty good. The feasibility seems to be pretty good, and I'm happy to report that the outcomes seem to be pretty good. So the other key thing that we do in this particular experiment is do something while the participant is receiving TMS. So a relatively, I mean, it's not really new, but a relatively new thing that people are talking about in the literature is task-dependent plasticity. So we know from our preclinical friends that a primed neural circuit has much higher plasticity potential than an unprimed neural circuit. And you can verify that statement through a conversation about dendritic spines or sort of treadmilling rates or cocaine self-administration profiles. We know that engaging the neural circuit in the task makes it much more likely to manipulate that task or the memory or whatnot. And so while people are receiving TMS for addiction, again, our idea is to dampen down the brain response to cues. So we have them imagine their cocaine cues, or in this case, their smoking cues or their alcohol cues, whatever it is of interest. And so we go through this standardized script induction of the last time that they used, describing the positive attributes of the experience. And we have them view the images while we're receiving the TMS. We have them keep their eyes open as well, which is sometimes a challenge, but it's generally pretty good. Okay, so what are the results? Well, so initially, again, a lot of people in the field thought I was a little crazy to do this over the ventral medial prefrontal cortex, because they had tried it maybe on people, and it can be very painful. But the secret is in the ramp. If you start low, you can actually bring people up to a clinically relevant dose. And so we started small with just a small pilot study of 11 individuals. These were cocaine-dependent individuals. And what we found, in this case, we did interleaved TMS, fMRI, as our probe, CTBS, an LTD-like form of TMS to the frontal pole, and then real or sham within subject, and then TMS as a probe afterwards. And what we found were specific decreases. This kind of blew my mind. A specific decrease in evoked bold signal in the nucleus accumbens, ventral striatum, and in the orbital frontal cortex. This is phenomenal. To anyone that's ever actually looked through their neuroimaging data on a subject-by-subject basis, you would be surprised to see that we actually found this in the majority of the individual participants. So that was kind of cool and maybe surprising, but it's a small study, and there's only so much we can learn from a small study. So we moved to a larger study where we had 49 individuals, 25 of whom were chronic cocaine users with use histories of about 15 years or so, and 24 of whom were chronic alcohol users. The reason we chose alcohol users was initially because our cocaine users had really strong alcohol use histories. And so in some ways, we were trying to use them as a control group. But it turns out, of course, that they're actually very interesting in their own right. So in the cocaine users, this same design, we see here that, so in the sandwich design, using TMS as a probe and then as a modulating tool and a probe again, that CTBS within subject sham controlled shows specific decreases in evoked bold signal in the ventral medial prefrontal cortex, the cingulate, the striatum, and the insula. So if you see here, these are all very classic, even the subgenual cingulate, these are all very classic limbic areas that we think of when we think of psychiatric disease and substance abuse and craving. That was very exciting. And these are the actual data here. So our significant clusters were here in the caudate, the accumbens, and the cingulate, as well as some precuneus and posterior parietal cortex. And that is just published. And in fact, we actually see a similar pattern in our alcohol users, which I have here. The alcohol users also have a decrease of evoked bold signal in sort of the ventral surface of the brain, medial prefrontal cortex here, as well as in some insula and the posterior inferior frontal gyrus. So again, here you see these sort of ventral aspects of the brain changed by CTBS. Then a very bright postdoc in my lab, Tanisha Kearney-Ramos, analyzed the data where we did cue reactivity immediately before and after the same paradigm. So again, in one case, so the basic scientist in me really likes the Interleaved TMS data because that's the principle, that's the fundamental mechanism we're looking at. But of course, if we're trying to develop a clinically relevant procedure, we probably want to look at cue reactivity because we know that cue reactivity leads to relapse. We could have done attentional bias. There's many tasks we could do, but task-specific activation is probably very important. And what Tanisha found, briefly, is that in cocaine-using individuals, real TMS specifically changed the connectivity, task-based connectivity to cocaine images in our cocaine users. And this was also true in the alcohol users. And I'll unpack this for you in the last few minutes of the talk. So what she did is using psychophysiologic interaction, so PPI tasks. This is a way to measure functional connectivity, but in this case, we're looking at task-modulated functional connectivity. So how is it that your brain oscillates between looking at neutral pictures and then is basically brought online when you see cocaine pictures and then offline when you see neutral pictures again? So it's task-modulated connectivity. She did a region of interest analysis, and she compared the blocks where individuals were looking at cocaine cues versus when they were looking at neutral cues. And what she found were in the cocaine users, baseline functional connectivity was high when they're looking at cocaine cues before they received TMS. After they received real TMS, but not after they received sham TMS, so these are real TMS-specific effects, there was a significant decrease in connectivity between the medial prefrontal cortex and the caudate and the putamen and the insula. These are tremendously exciting data. I'm very excited for her because as you may recall from the initial slides, these are the same brain regions which we know are associated with relapse in cocaine users. And so these data really do suggest that in cocaine users, we can apply CTBS to the medial prefrontal cortex and potentially make the brain look like the brain of somebody who won't relapse to cocaine. So that's pretty cool. Okay, and in our alcohol users, of course, data is good, and then if you can generalize it to another drug-using class or a whole new population, it's even better. And so even more excitingly, she found the same pattern was true in the alcohol users. So the alcohol users, their engagement of their brain, of the medial prefrontal to the striatal system when they see alcohol pictures, was specifically decreased following CTBS administration, but not sham. So it suggests that CTBS may be transdiagnostically appropriate tool to change the brain response to drug cues. And what we didn't find was a change in self-reported craving. So why is this? Well, we're not too worried about it because self-reported craving isn't really a super robust measurement. And in fact, generally, you need a lot of sessions in order to get a behavioral change. And so the last step that I'll talk about here is the newest study that we're doing, which is ongoing, and it's looking at sustainable changes in brain reactivity to cocaine cues in treatment-engaged individuals. So like I said, this is a This is a single-site, a double-blinded, sham-controlled cohort study in individuals that have enrolled in an outpatient treatment program where they're largely receiving cognitive behavioral therapy for 4 weeks, 28 days. When they enroll, we randomize them to receive 10 days of real or sham CTBS, and the sessions occur typically before their outpatient CTBS CBT sessions. We apply the TMS between week 1 and week 4 of their outpatient treatment, and then we follow up 1 month and 2 months later, and we're looking at cue reactivity at these different time points. Well, right now, the study is still blinded, so it's hard to know what to tell you all, but it's very exciting. And what I can say is that since it's blinded, we have the ability to kind of look at Group A and Group B. And it does, in fact, seem as though Group A and Group B are starting to separate. We're about halfway through our data collection for the study. So if we're lucky, they're separating in the right way. But, you know, they may be separating in the wrong way. And that's honestly the fun thing about science, right? Either way, we're going to learn something. And so hopefully the data will be ready to present at ACNP in this December. And so I'll have more for you all then. And so I just kind of want to sort of end with a little inspiration. So the theories which I've expressed to you here may appear to be, I'm paraphrasing because I can't read, may appear to be very chimerical, but they really are extremely practical. So practical that I depend upon them for my bread and cheese. You know, all the time, especially those of us in the extramural program, we've become very good at using all the buzzwords of language, like translationally relevant, evidence-based treatment. And they sound good, and we believe it. But in this case, like, we really might be able to make a difference. We really have a lot of really strong preclinical data that suggests that these circuits have a causal effect on drug self-administration. And we have more and more rigorous sham-controlled studies in cocaine users with TMS that show we can move these circuits. And they're sort of buttressed by this huge body of knowledge that suggests these circuits are important to relapse. And so I think with a community of us working together, we really could bring this to sort of an FDA-approved treatment for cocaine dependence sometime maybe in the next 5 to 10 years. That's very exciting since there's currently no FDA-approved treatments for cocaine dependence at all. And so our vision is to be able to deliver our TMS as a translationally relevant neural circuit-based brain stimulation treatment for cocaine dependence in the next 5 years, if we're lucky. There's lots of questions out there. There's lots of opportunities for you guys and everybody out there viewing to make a difference in the field. Tons of questions. And just like the depression field is still working on all of these optimization protocols now, a decade after FDA approval, and many years after Medicare and Medicaid have been reimbursing patients for it, I think we too, as substance-dependent researchers, have a lot of time to work out all of these details. And it's very great, and it's exciting for our future. We just need more people to participate. So come on, join us. Okay, I'm going to skip through this because I think we're out of time. But individual variability is very important. There's some suggestion that various factors contribute to it, and Riving and Zeman have done an excellent review article talking about this, which I highly recommend. Okay, and finally, this initial cascade of evolution of addiction, which I talked about, the vulnerable, reward-seeking individual, and then the habitual drug-taking behavior that manifests. Those two different phenotypes, or different portions of the addiction spectrum, may in fact respond better to one form of TMS stimulation than the other. It may in fact be the case that individuals that have more reward-based mechanisms that drive their cocaine use are going to respond better to medial prefrontal cortex stimulation. And individuals that have more habitual-related reasons for drug use, potentially like we think about with cigarettes, and smoking, there's been great multi-site trials with TMS as a tool with the brain sway coil of developing treatment for nicotine. And in that case, they're stimulating the sort of more of the sort of motor, habitual, executive control areas. So that will be really exciting to see as well. And then one last thing, as last word of inspiration. So John F. Kennedy gave this speech a while ago, and we thought at a time in which it was sort of very odd to think of going to the moon. We had never really maybe left the planet a little bit, but we certainly hadn't done anything as great as putting a man on the moon. That seemed so crazy. And I think to many of us it seems crazy to actually be in these rooms on a daily basis, doing the job we do every day, and actually think we can make a difference. But we can. And so listen to his words. We chose to go to the moon. We chose to go to the moon in this decade and to do other things, not because they're easy, but because they're hard, because that goal will serve to organize and to measure the best of our energies and skills, because that challenge is one that we're willing to accept, one that we're unwilling to postpone, and one we intend to win. And so with that, I wanted to say, I have a lot of friends that do TMS, and I have a lot of friends that try and then they get discouraged because it's a gigantic parameter space and no one knows what to do. But don't be discouraged. We have the opportunity to really come up with new treatments, and there's a lot of evidence. And I think we can make a difference. And that's all. These are all the great people that help me all the time.
Questions on the chat window?
Okay. Do any of you all in the room, or does anybody in internet land have a question? You can apparently type them into your window, and maybe I would see them in the chat window. Do you all have any questions? Yeah. Uh-huh. Why do you pick CTBS? Why do you pick usually CTBS is given lower intensity. Yeah. I mean, it's not going to be probably the same. Yeah. But why do you pick this question? Yeah. Yeah, absolutely. So that's a great question. So the question was, why is it for CTBS that we're picking such a high intensity? So let me go back to that slide. And the answer, here's our pulse parameters. So we're using 110% of resting motor threshold. And I should say, so the Downers group uses a resting motor threshold for the foot when they stimulate the medial wall. And that's actually because they're actually not too far from the motor cortex of the foot. So that actually seems pretty smart. It's really hard. I'm not very good at getting motor evoked potentials from the foot. And I sort of was trained by people that use the arm. So we use the arm or the hand. And we use 110% mainly because of the distance. Mainly because the distance from the frontal pole to the surface of the skin is much greater. So that's really the reason. Another related question there is, why did we use 3,600 pulses initially? So the reason we used that, again, we knew less, so we used that because we wanted to use theta burst stimulation. It's biologically relevant. It's a natural endogenous brain rhythm that's involved in learning and memory. So that sounded great. We wanted to use continuous theta burst stimulation. But because I was changing the frequency, to the burst frequency, I wanted to minimize the number of other parameters that we were giving. And so because clinically, to treat for TMS, people often use about 3,000 pulses, we wanted to keep the pulse number approximately the same, somewhere between 3,000 and 4,000 pulses. But CTBS kind of off the shelf, CTBS kind of comes in 600 pulse units, so to speak, right? So we needed to do a multiple of 6, and so we chose 3,600. Yeah. Thanks. Mm-hmm.
So there's a question, do you study how to maintain the effects of, do you adjust the stimulation intensity to the depth, or do you apply 100%?
Yeah, that's a great question. So the question was, do we adjust the stimulation intensity based upon the distance between the scalp and the brain, or do we apply 110% to everyone? That's a great question. The answer is right now we apply 110% to everyone, and then we use the distance between the scalp and the cortex as a covariate in our analysis. So that's what we've been doing, but I actually think it's completely reasonable to dose the TMS based upon the distance, and I actually could see that the field might potentially want to move in that direction in terms of translating this to a clinical treatment, right? Refining it for your individual.
And do you study how to maintain the effective TMS for a longer time, i.e. maintenance?
So that question was, do you study how to increase the durability of TMS over time, i.e. maintenance protocols? So, so again, in addiction, we're far more immature than depression, and so we really are at square one. I do know that there's actually a lot of emphasis within depression literature all around the world. Actually, groups are looking at durability of treatment effects. So no, we are not doing there because we're not there yet. We aren't quite sure that we can actually change the disease yet. But once we get there, we will definitely think about maintenance. Okay, great. Well, thank you all very much. Okay, I think that's good. Yeah. Okay.